The second part of our biotechnology notes are all about different ways that scientists can actually study the DNA that you would find in a gene or a genome. So looking at things like the differences in sequence between people, the actual sequence of a gene, and stuff like that. Now, one of the oldest approaches that we still use today is called gel electrophoresis. And this is something that I hope will sound a little bit familiar because we do cover this briefly in the freshman course. And this is where we're trying to separate things out. So I might spell this wrong, but the goal of electrophoresis is separation. So previously you looked at PCR, and the goal there was amplification. The goal now in this electrophoresis process is separation. So we're going to use a gel called agarose to kind of act as a strainer for us. And if we move DNA through it, we will be able to separate out different pieces of DNA. So first thing we do is we take a DNA sample and we're going to cut it up with these things called restriction enzymes. And we'll come back to that here again in a minute. Restriction enzyme will chop your DNA up into different size fragments. So after we expose DNA to a restriction enzyme, we might have a couple different sized pieces of DNA. Then you'll take your mixture that's in your test tube, load it into a well in the gel, which is like an opening at one end, submerge the whole gel into some saline, basically some salt water, in a chamber, and then run an electric current through that chamber. Now what this is going to do is it's going to move your DNA because DNA has a slightly negative charge. So if you got this DNA in here that's slightly negative, and then we run a current in our chamber that runs from negative to positive, then the DNA will actually move toward that positive charge. Now, the reason it separates out is that these fragments are different lengths, right? And the larger the fragment, the harder it is to move through this gel sieve or this gel strainer. So we get something, this is a much better picture than what I was drawing on the last slide, where we load our DNA into the gels here at this location, and then we run this electric current through the gel. And what happens is that the DNA will make its way, at least in this picture, from left to right, but the bigger molecules, the bigger fragments, won't go as far, so these are larger, as smaller fragments, because the smaller fragments have an easier time moving through that gel. What you end up with at the end is a series of lines, where each line represents a fragment of DNA that came from our original sample. And now those fragments are separated out by their size. Now, the question becomes, why would this help us, or why do we want to use this for something? First what we want to say is that a big sample of DNA is basically like too much for electrophoresis to handle. In other words, if I have a really big DNA sample and I try to load it into an electrophoresis gel and then I run it, what's going to happen is I'm going to have so many lines that the lines are just going to overlap each other. And what we usually end up with is something that looks more like a smear, is what we would call it, than separated out lines. And I'll show you an example of that here in a minute with a picture that's in here. So when we do electrophoresis, we're usually either going to use something that has very little DNA, so like a bacterium, or we're going to only use a particular target sample. So maybe we're only going to use this little area of DNA from one of the human chromosomes. And that's what we'll be doing in our lab in classes. We're going to look at a particular gene sequence from one of your chromosomes. Now, the reason that this is useful has to do with what a restriction enzyme is. a restriction enzyme cuts DNA, or kind of clips DNA at a specific sequence, called a restriction site. So we got a specific sequence called a restriction site. And any time that the restriction enzyme finds that sequence, it will cause it to be cut. So I'm making this up. This is not a real one necessarily, but let's say that you've got a restriction enzyme, and every time it finds this sequence, this is our example, every time it finds this sequence, ACCA, it causes a cut between the two C's. And of course, this is a double-stranded thing, so it would be T, G, G, T on this side. So, it causes this cut through that strand. And what makes this interesting is that this sequence is going to be found in different places in different DNA samples. In other words, all of us will have this little restriction site in different places within our genetic complement. So let's say we take different samples, right? We got this sample, we'll call this one, this is a sample from Jim, and this is a sample from Bill, and this is a DNA sample from Judy. And maybe Jim has, this restriction site just in one place, and it causes a cut right there. But Bill has this restriction sequence in two places, so his DNA gets cut here and here. And then Judy actually has this restriction, this ACCA sequence, show up in 3 different places, or let's say 4, right? So now we've taken this same segment of DNA from these people, but when we expose it to a particular restriction enzyme, each person's DNA will cut a little bit differently. Notice now that Jim has 2 fragments that are pretty long. Bill has 3 fragments of DNA, all of them a little bit shorter. And Judy would end up with 5 different pieces here, all very short. All right, so that's the point of this last note here. Everyone who varies will have a different set of fragments, and therefore a different set of lines. If we run these DNA samples in an electrophoresis gel, we're going to get something different for each person. So let me come up here, clear me some space real quick. And now let's say we're going to run this DNA. So we've got Jim, and we've got Bill, and we've got Judy. And so we load their DNA into these wells. Remember that Jim made 2 pretty large fragments of DNA. So when we do electrophoresis, maybe we get 2 lines like that, right? And that's his first fragment and his second fragment. Bill is going to have 3 fragments, and all of his were smaller than Jim's. So smaller means they'll move further down the gel. So maybe Bill ends up with a pattern like that. And then Judy, remember down here, gave us 5 segments, all of them very small. So maybe she gives us, yellow is hard to see, a pattern like that. Now here's what's cool. Every time we expose Jim's DNA to this same restriction enzyme, we're going to get this pattern. And so now we have basically what's referred to as a DNA fingerprint. A person's DNA will react the same way every time we hit it with a restriction enzyme. Now when we go in and do really what we just did on the last slide, which is compare different people's pattern, what we're looking for technically is referred to as a restriction fragment length polymorphism, or sometimes referred to as a RIF Flip. Now this sounds like a mouthful, restriction fragment length polymorphism, but if we break it down, it's not crazy. Polymorphism is a difference in DNA sequence. So if we look for restriction fragment length polymorphisms, a restriction fragment, all right, so the first part here, that's just a DNA fragment. right? And then we got length. We all know what that means. And polymorphism means a variation. So what we're looking at here is different length DNA fragments, right? RIF Flips. Now, this can be really useful in forensics, right? Let's say that we have here, 3 suspects for committing a crime in this upper gel. And then we have some crime scene blood samples. So, here's our samples, A, B, C, and D. Notice that line 3 and line C match. That's an indication that this suspect number 3 was at the crime scene, right? Because we're going to get the same pattern every time we run their DNA. It's also useful for determining relationships between people, although, as I put down here, we do have sort of more advanced and more efficient technology today that we can use for this. But this is the classic way to do it. Also, in confirming if you have a certain genotype. A lot of times this might involve a disease or a disorder like sickle cell, or in the case of like the lab will do in class, just looking to see if you have a particular genotype. And all we need is a couple of controls, right? In this situation, we have a control for normal blood allele and a control for sickle cell allele. So then we just run our unknown person here in our third well, and we see if their pattern matches either of those patterns. And the second thing I'll mention, although this is not super critical, is called Southern blotting. Southern blotting is named after a scientist named Southern, not the direction on a compass. And this is a way to examine a really large DNA sample, but focus on just a particular gene of interest. So it's like a combination between electrophoresis and then also nucleic acid hybridization that was mentioned in the first part of the notes. Basically, here's that picture I mentioned earlier of what happens if you run electrophoresis with with lots of DNA. All right, you get these smears, these streaks. But let's say that's what you've got, and you really want to be able to focus on a certain gene. Well, what you can do is you can put in there nucleic acid strands that match up to or would base pair with what you're interested in seeing, and then you can end up with an image that's more able to be compared, like you see here. So let's take a look at how this works, but again, don't get too hung up on this one. This, I know this is not nearly as important as just electrophoresis in general. So basically, you do everything you need for electrophoresis, right? So we take some samples, we hit them with the restriction enzyme, we run electrophoresis. Then what we have to do is transfer our samples basically from our gel over to our blot, which is a different type of material. And once we've done that, we can take that blot and put it into some liquid along with some radioactively labeled probes. And then they will bind to those particular gene sequences that we're looking for and light up or something like that, so that now when we look at our blot in the end, we just see the lines we were interested in instead of all these lines that we saw in electrophoresis. This is really not important to understand how this is done. Now, the other kind of technique that we do want to know a little bit more about is a way to figure out what the actual sequence of a gene is. And so we haven't talked about that yet, right? Like, you've got a gene, but the question is, how do we figure out what the sequence of A's, T's, G's, and C's are in that gene? This is something that baffled scientists for quite a while, until a man named Fred Sanger came up with a way to do this, which is called the dideoxy chain termination method, or much simpler, Sanger sequencing. And So I put here, Fred Sanger has 2 more Nobel Prizes than I do. So this is a double Nobel laureate, which is pretty amazing. One of kind of the people I look up to a lot in biology because he did some amazing science. But basically, he was the first one to figure out how we could take a gene and then tell the actual order of nucleotides that are in there, so the actual sequence. And he found a way to do this in an automated way, so with a lot of help from a computer, but in a way that's still very physical in nature. So in Sanger sequencing, which is really complex, as I put in here, this is the simplified version, which is scary because this is still going to be, I think, tough to grasp. But basically what we do is we take a DNA sample and we put it into a test tube, right, with everything we might need to do DNA replication. So we're going to take a DNA sample, put it in a test tube with DNA polymerase, with helicase, with all those enzymes, with A's, T's, G's, and C's, so that theoretically we could almost do PCR, right? We could almost copy that DNA and make copies of it, but that's not our goal here. And what we're going to do is we're going to add one more ingredient, which are these things called fluorescently tagged dideoxynucleotides, and those are pictured here in this illustration here. So a dideoxynucleotide is one that is special because it has a slightly different structure, but what that little bit of a structural change does is if it is incorporated into the DNA, it will stop replication. So if you put in one of these dideoxy A's, then right then you will stop replication. Now what happens is in your test tube you get the conditions just right so that you will do replication. But what ends up happening is whenever the strand randomly uses one of these special A's, T's, G's, and C's, it'll stop replication of that strand. So what we end up with is a test tube with a whole bunch of different strands, each of them basically one nucleotide longer than the others. So once we get that, again, we're still just talking about a liquid in a test tube, right? So what we can do is we can use some advanced technology here, use some computers, use some lasers, and separate these DNA strands out by length. Now remember, these are fluorescently tagged dideoxynucleotides, which means they're going to light up a certain color. So when we then look at this separated out column, which is what this would be, we can actually figure out what the sequence of bases are. And so we'll have our origin, our starter that we're aware of. So in this case, it's this TTGT. The smallest strand we would then notice has a pink end on it, at least in this picture. And we know that the C's, the dideoxy C's, are tagged pink. So now we know that that fifth base is a C. All right, so I'm writing it over here. Now we know that fifth base is a C. Then we look at the next longer strand, and it's got a green end on it. Well, green is the tag for G, so now we know the next base is a G. Then we look at the next longest strand. It has a purple end, and purple was the color for A, so now we know that's A. And if we do this for the whole thing, we can end up telling what our sequence is. Now I know this makes as much sense as my children wearing underwear on their heads. So we will do an activity in class to walk through Sanger sequencing that I do think will help you quite a bit in understanding how we break this down and how we actually can use this. Now we got a couple other things just to throw at you. These are mentioned in the AP framework. I don't know that they're going to be a big deal. In fact, I don't know that I've seen a question about them yet on the AP exam in May, but you never know, and it might kind of be the thing where they just mention it in a question and it's not super critical, but at least this way you will have heard it before. So a lot of times we want to know if a gene is being expressed, and we want a test or an analysis to be able to kind of quantitatively say that yes, this DNA is being expressed in a cell. And one of the ways you can do that is to look for mRNA. basically, I don't know if we've said this yet, you know, if transcription happens, right, if we go from DNA to RNA, then that pretty much commits us to making proteins and doing translation, right? So if we make RNA, we can safely say that that gene is being expressed. So basically, what we can do is we can search for RNA in a cell, and if RNA is there, then we say that that's indicative of gene expression. And the way that you can do this and look for mRNA is called northern blotting. Now, northern blotting is not named after a scientist. It's a joke, basically, right? Scientists do have a sense of humor sometimes. We had southern blotting, so they made this thing called northern blotting. And northern blotting is a lot like southern blotting that we saw a few minutes ago. The difference is that you do electrophoresis, but you do it using mRNA samples instead of DNA samples. And then you use blotting, and you use DNA probes to recognize or find out if there is mRNA that match those sequences. How this is done is really not important. The main thing that I would take away from it, I think, is this allows us to do gene expression comparisons, which leads to some cool research, right? Maybe we can look at how much we tend to express the insulin gene versus how much a cow tends to express its insulin gene, or one of the other great apes, right? How often do we express insulin genes versus chimpanzees? You can also look at different people or something like that, right? Do older people express more of their more or less of their immune system genes than younger people? Things like that, and this would allow us to not only just compare, well, you know, we know that if you have the genes, you're going to have them for life, but the question is, how often do you use those genes or express those genes? And northern blotting will allow scientists to sort of make comparisons of that. But again, I don't see this being a big sticking point moving forward. One last thing, which is pretty much the most modern thing we're going to mention, is something called RTPCR. So polymerase chain reaction, PCR, remember, was a way that we copied genes or we amplified DNA. Reverse transcriptase PCR means that we're going to do PCR, and we're going to do that to amplify a gene, but we're going to start with RNA. So essentially, if you think about it, this is reverse transcriptase is an enzyme that does transcription in reverse. So we're going to get some mRNA, and we're going to turn that back into DNA. And then we're going to use PCR to copy the DNA. So this is also a way to kind of look at expression a little bit, right? If there is RNA present, it means those genes are being expressed. So then let's go back to the DNA and copy the DNA and take a look at what that gene is that is being expressed. And we'll talk a little more about reverse transcriptase later, because some of you might already kind of recall this from biomed. This is an enzyme that is present in viruses who actually do this and go from RNA back into DNA. All right, so probably by the time you're watching this, we have already started in class doing some biotechnology stuff. But the way to really learn this is to do it. So we're going to try to give you as much opportunity as possible to do some of these things in class, so that hopefully they make sense, and when you run into them on a test later, you'll be able to talk about it. Thanks for listening.